We warmly welcome everyone. Our names are Adam and David Tarnoki. We work as assistant professors at the Semmelweis University Medical Imaging Center, as well as in the National Institute of Oncology in Budapest, as well as we lead the Hungarian Twin Registry. In our lecture, we would like to summarize the most important findings of imaging twin studies in Hungary. In the first part of the presentation, we would like to introduce the topic, as well as we would like to first talk about the heritability of the carotid artery phenotype. So this is the main neck artery which supplies the brain. These are the cardiovascular imaging methods in twin studies. This is the different imaging methods are explained here in this slide. The first one is the X-ray and the computed tomography. So the common link between the X-ray and the CT scan, they both work with X-ray exposure. However, the CT has a high resolution, and this is a much higher radiation dose, since here the X-ray tube is rotating around the patient. However, there are low dose protocols which help the screening studies will require less radiation exposure, such as the low-dose CT scans of the lung. Moreover, taking into account then the X-ray and the CT has a radiation exposure, especially the CT scan. The twin studies in this topic are usually retrospective. The second topic is the ultrasound and the MRI. So both of them, both the ultrasound and the MRI, have no radiation exposure. These are widespread imaging techniques. The MRI is especially popular in neuroradiology, and new ultrasound and MRI applications are available thanks to the developing technique. The combination of imaging phenotype with the epigenetics, microbiome, and other studies, such as radiogenomics, is also emerging field. 125 million twins live worldwide, and among them 130,000 twins live in Hungary. And Hungary is in heart of Europe, in Central Eastern Europe, and this is the map of Hungary. 10 million people live here, and 5 million Hungarians live around the country in the neighboring countries, as well as in the world. The number of twin births in Hungary is represented on this slide. Since 1970, the population of under 45 years and older, approximately 65,000 twin births occurred, approximately 130 and 170,000 individuals. In the past 40 years, in average, 1,500 twin births occurred annually in Hungary. You can see here on this slide the incidence of the twin births, the single birth ratio as well as the twin birth ratio. As you can see here, indicated with red side, the twin birth ratio is increasing mainly since the beginning of the 1980s. Here are the number of twin births in Hungary and Budapest. In Hungary, as you can see, there are several counties where the twinning rate is higher. However, in some of them, the twinning rate is less. This was in 1990. However, in 2012, the central counties show a higher twinning incidence. It can be due to the higher population in this area. On the right side, you can see the map of Budapest and the various districts of Budapest with the twinning rate. The dark color indicate higher twinning rate. In 1990, you can see that there were less districts with higher twinning rate compared to the map in 2012, where the twinning rate is almost very high in almost every district. So this is the Central and Eastern European map, and you can see that in Hungary we have the only twin registry in the neighboring countries, compared to the neighboring countries, where only twin clubs and associations are available. And twin studies are also rare in the neighboring countries, so we can be the number one in the Central Eastern European region. On the right side, you can see the consistency of the current voluntary twin registry. Currently, we have 1,050 twin pairs in the registry. Half of them is identical and half of them is dizygotic. And we have some triplets, quadruplets as well. And we have a female dominance. The mean age is in 34 plus minus 22 years. However, we would like to turn to a population-based registry. For 5 years, we have been working on this on the foundation of the population-based twin registry, and we will mail 130,000 twins. This is the total number of twins living in Hungary based on the statistics, and we have a new website as well. Here is the new website. So this will be there will be a place where the twins can register here via the website and give that data for the registration. However, there will be forum, chat, news, question and answers, and publications as well. So this will be a widespread website. And here is the turning mechanism to the population-based twin registry. So we will mail these postcards to twins and asking them to register online. However, for those who have no internet access, especially the elderly twins, there will be a call center and we can call them back, and also a mail contact. However, for those who didn't answer and or register via call center and mail, we will mail them the questionnaires and they can complete the questionnaire. However, those who register online, they can complete the register or the questionnaire already on the website. So this is the team. So some of them are twins themselves, such as Julia Metaki and Dora Meliher. They are also twins themselves, and we have also many PhD students and student scientific researchers as well. Dr. Litva is the twin statistician. So in the first topic, we would like to talk about atherosclerosis. Most of our studies are based on this process where the wall of the arteries are beginning to thicken, and there will be a plaque development in the wall. So first there is an endothelial dysfunction, and the wall of the arteries will be stiff. This can be measured with various techniques. Then the wall will be thickened, and that is the so-called Intima media thickness, the IMT. That's the thickness of the wall in millimeter, and there will be a plaque developed. So we were investigating each part of this process. So first I'd like to show you the Intima media thickness, which is considered as abnormal if it's thicker than 0.9 millimeter. That's a direct marker of the atherosclerosis, and it can be measured by ultrasound very nicely. And in our twin sample, which was published in various journals, we could find that the monozygotic twin correlation is higher compared to the dizygotic twin correlations in the common carotid artery, as well as in the femoral arteries as well, and we could show a moderate heritability based on the results of the twin study. We also follow up the twins for 5 years, and then for 5 years in Hungary and in Italy, and then we investigated and measured the same twins 5 years later. So this is the with ultrasound. This is the carotid intima media thickness, and we calculated the intraclass and cross-twin cross-time correlations according to zygosity between the 2 time points, and we found that the genetic contribution to longitudinal stability is moderate for the carotid IMT based on the results. Here you can see the various standardized variance components between the so in the first visit the heritability was 32 percentage, in the second visit it was 58 percentage, and so this was a moderate heritability. So we also investigated the plaques in the carotid artery, and we found that the monozygotic twins correlation is much higher compared to the dizygotic twins concerning each characteristics of the carotid plaques. So the presence of the plaque, the genesis is the type, the size, the sidedness, the number of plaques, and the presence of the plaque, exact presence of the plaque in the carotid artery, and it indicated a very high heritability. Published in Stroke. Concerning the femoral plaques, we also investigated the same characteristics, so the presence of the plaque, the type of the plaque, the sidedness, and the number of the plaques, and we found very similar findings compared to the carotid artery. So we found moderately high heritability based on the result. And afterwards, we were interested whether the carotid and the femoral intima media thickness and plaques are presented in both twins as well as in both locations. And we found that the average IMT was a little bit, so the wall thickness was a little bit higher in the femoral artery, as well as the plaque prevalence was higher in the common femoral artery compared to the carotid one. So it was very interesting to see that there were some subjects which had no carotid plaques, however the femoral plaque was present during the ultrasound, and we analyzed then also in details that in half of the twins who had plaques was the plaque presented in both locations only. So the difference is more pronounced looking at the fibrolipid, the soft plaques. So this was much higher in the femoral artery, much more frequent in the femoral artery compared to the carotid plaques. And this can be seen here. This slide shows a case of a carotid plaque, a calcified carotid plaque in a 69-year-old monozygotic twin pair. They are both ladies, and it's interesting to see that both of them has a calcified plaque on the carotid artery. You can see that on the top, almost in the same size and same location. However, on the femoral plaque, the twin A, so the firstborn twin, has a big femoral calcified plaque. However, the other twin, the twin sister, you can see it on the right side, has also a plaque which is currently a soft plaque. So this slide shows that, that especially in the carotid artery, both twins have calcified plaques indicating a heritable background. This was the femoral plaque of the same twin pair, and we found an almost identical femoral calcified femoral plaque here. So you can see almost the same size. We measured that in both members of the twin. So it was interesting to see the femoral plaque was identical. However, the carotid plaque was not the same. The firstborn twin had much larger carotid plaque than the secondborn twin. This is a 62-year-old identical twin pair. They are also both women, and you can see in the carotid artery, and which is marked with this green sign, a soft plaque. However, the other twin had a mixed plaque with calcified and soft parts as well. So it was interesting to see that it's not the same, so this was also indicative of the negligible genetic background of the soft plaques. Regarding the femoral plaques, this is also an example for that. That's also an identical twin pair, same location in the femoral plaque. The firstborn twin had only a mild wall thickening. However, the second twin on the same location had already a mixed plaque. With a mainly soft component. So this is also indicative of the negligible genetic background of the plaques. In the second part, we'd like to talk about the heritability of the coronary diseases, epicardial and abdominal fetus, and we would like to also talk about the disease of the Willis and the presence of the Willis circle and some lung diseases. So we concentrate on these two areas. So regarding the heart of the twins, this was the first twin study in the world where we investigated the heart of the twins using the coronary CT angio. And this is the example of one identical twin pair and the 3D reconstruction of the heart with the virtual image of the main coronary artery, the left anterior descending artery, and you can see here also the reconstruction of the artery indicated the various plaques in the wall. And this was very identical in the twins. However, you can see that the anatomy of the artery was not the same. Looking at this branch, it was much stronger in the first born twin. However, it was much smaller in the second born twin. Regarding the coronary plaques, we found that the calcified plaques were more similar in identical twins, indicative of the genetic background. However, the soft plaques, which are more dangerous regarding the myocardial infarction, they were more dissimilar in identical twins, indicating an environmental background. Regarding the aortic root dimensions, we also analyzed the aortic roots of the twins in almost 200 twins in the same study, and we found that the aortic root dimensions are determined by genetic and environmental effects as well. But as you can see here on the right side, regarding each segment of the ascending aorta, they were mainly determined by genetics. And we found that the heritability was high for both epicardial and abdominal tissue quantities. This can be seen here on this table, that epicardial and subcutaneous and visceral fetus were, and genetic effects predominance over environmental influences contributed to the phenotype of the EAT quantity. And similar to the abdominal, subcutaneous, and visceral fat volumes. Common and specific genetic effects may be involved in the heritability of all three adipose tissue quantities. Regarding the brain and brain arteries, in another Hungarian twin study, we analyzed with MRI the morphological variants of the circle of Willis. This is in the base of the brain, and the various variants are very common also in the general population. We found that there are environmental rather than genetic factors in the background of the morphological variants of the circle of Willis, indicating that the monozygotic twins were mostly dissimilar in the presence of the certain variants. So the role of the stochastic and in utero factors arise. Here you can see the results that mostly the dizygotic twins were more similar compared to monozygotic twins, which indicated that there was no genetic background. Regarding the lung, that was a low-dose CT study in Hungarian twins involving twins having chronic obstructive lung disease, COPD. So in these previous twin studies indicated that COPD is heritable in general based on lung function. However, there were no morphological twin studies involving twins and involving twins who underwent CT scans. So we involved some COPD discordant and concordant monozygotic twin pairs who underwent a low-dose CT scan, and we found that the results of similar lung parenchyma and small airway changes, indicating in monozygotic twins a genetic background regardless of smoking state. Here you can see the volume reconstruction images of the lungs and of monozygotic twins in three monozygotic twins and 2 dizygotic twins. As you can see here, in the first row there's the firstborn twin, in the second row you can see the secondborn twin. So it's very similar how the lung morphology looks like in monozygotic twins compared to the dizygotic twins in genetic background. Regarding the air trapping distribution, we found also similarities in monozygotic twins compared to the dizygotic twins. These are air trapping, these air lungs in the lung, some areas where the lung remains there by exhalation of the air because of the disease of the small airways. This is the emphysema distribution in monozygotic and the dizygotic twins, which was also similar in the monozygotic twins. In the third part, we'd like to talk about the microbiome and the association of the microbial diversity in the gut and the various cardiovascular imaging phenotypes and the results, most recent results of a twin study. So this is thanks to the collaboration of various colleagues in MLS University. So the composition, well known, the composition and diversity of the microbiome in the gut influences the development course and therapy of various diseases such as the cardiovascular diseases. And this can be influenced by lifestyle changes in parallel. The metabolic syndrome was known to be influenced by diet, altered metabolism, decreased microbial diversity, and which can be administered with appropriate pre- and probiotics, and short-chain fatty acid production is influencing that. The atherosclerosis is an infection, immunity, bacterial products, and dietary metabolysis influence these influences its development. So our aim was to explore this relationship between microbiome diversity in the gut and the cardiovascular phenotypes and other diseases. So we involved 114 twins to the study based on selection criteria, and for example, those twins who had carotid and femoral atherosclerosis based on a previous twin studies were involved, and those twins were discordant for certain diseases, as well as we asked the twins to be involved for ultrasound examinations, and we analyzed the arteries by special program. This is the artery analysis program where we analyzed the certain features of the atherosclerosis, such as this is the size of the plaque, the volume of the plaque was calculated in, as well as the automatic intima media thickness, so the wall thickness. So we asked the twins to cooperate in the study, and we asked them to send a stool sample where DNA was extracted, and various library construction was performed and analysis of the microbiome composition. And afterwards, we compared the studies of the carotid and femoral ultrasound examination and the questionnaires with the microbiome composition. So here you can see the twins of various what we found, the microbiome composition, including the phylum and genus taxonomy levels. Here you can see the twin number, the first and second born twin, and you can see on the right side the certain names of the bacteria and the comparison. And this was how the analyses were done, and the results you can see down on the lower image, where you can see the 10 most common genera in subject with increased IMT. So here we selected the monozygotic twins and those whose IMT was thickened on the carotid artery and those whose IMT was in a normal range. So, and here we found some bacteria in some bacteria such as the Faecalia bacterium, where we found a significant difference between those twins having increased IMT and decreased IMT. So in general, we can see that the genes can be influenced by the environment. This is what we are investigating. There's many other positive and harmful as well as variable effects from the environment, including the microbiome, which influences the epigenetic modulation and the genetic modulation of the phenotype. So heritability of environment, this is what we are investigating. So the genetics, epigenetics, and the environment influences the phenotype, what we will see with imaging technique. So this is the vision. So the genetic, the genomics, epigenetics, microbiome, and environment all influences that phenotype, which we can investigate using radiological imaging methods. So as summary, we would like to say that the modern X-ray, ultrasound, mammography, MRI, CT, and molecular imaging measures can be used to define these endophenotypes. The integration of imaging with twin research contributes to the heritability estimation of imaging phenotypes and assessment of the role of microbiome and epigenetics in disease development. With this, we would like to thank you for your attention.